We typically think of illness as an invasion. A virus or bacteria breaches our defenses from the outside, attacking our cells to make us sick. But cancer operates differently. It isn't an external attacker. The threat originates from within our own biology. To understand how this happens, we have to look at how a healthy body maintains itself. Throughout your life, your body survives by replacing older cells with new ones in a highly regulated, orderly process. This happens through mitosis. A single mother cell copies its internal DNA, the genetic instructions, and divides into two identical daughter cells. The DNA inside the nucleus acts as a strict manager, utilizing specific checkpoints to regulate exactly when division should start and when it must stop. Our health depends on this microscopic obedience. When cells follow these internal instructions, the body's tissues grow and repair themselves without spiraling out of control. The problem begins when the DNA instructions acquire a typo. This is a genetic mutation. If that mutation occurs in the specific genes that manage those cell cycle checkpoints, the cell loses the ability to govern its own replication. This breakdown involves two critical types of genes. The first are oncogenes, which act like the gas pedal of the cell cycle. When an oncogene mutates, it gets stuck in the on position. This drives the cell into constant, unstoppable replication. The second type are tumor suppressor genes. These act as the cell's braking system, designed to halt division or trigger cell death if they detect an error. A mutation here effectively cuts those brake lines. The cell is stripped of its natural ability to stop, even when it identifies its own genetic damage. Transforming a normal cell into a runaway biological threat requires both of these failures, an accelerator that won't release and a braking system that no longer functions. While some of these mutations are hereditary, many are caused by external factors encountered during our lives. These factors, called carcinogens, physically damage the DNA strand. If the cell's repair systems fail to fix the break, the error becomes a permanent part of the cell's code. This unchecked division creates a physical mass of abnormal tissue known as a neoplasm, or a tumor. A benign tumor remains localized. It is encapsulated within a boundary, meaning it pushes against surrounding tissue but does not invade or destroy it. Malignant tumors are different. They are aggressive, breaching their boundaries to invade and destroy nearby healthy cells. This leads to the most dangerous phase, metastasis. Individual malignant cells break away from the primary tumor. These cells travel through the bloodstream to establish new, secondary colonies in distant parts of the body. What begins as a microscopic genetic typo in a single cell can lead to a systemic failure as these abnormal cells proliferate and spread. At its core, cancer is a single human cell that has lost its programmed ability to stop dividing. Modern oncology focuses on identifying the specific proteins and pathways where this regulatory system failed. By identifying the exact genetic mutations that drove the breakdown, researchers can map the specific mechanics of an individual case. This allows medicine to move away from broad treatments that damage healthy and cancerous cells alike. Instead, clinicians are developing therapies that target the specific broken pathway of the cancer cell. Every discovery regarding these microscopic rules provides a new point of intervention, allowing us to use our understanding of cellular logic to disrupt the growth of the disease.